Great. Well, thank you so much, Anne, for the introduction, and to Andre and the other organizers for the opportunity to speak here today. So I'm looking forward to telling you about some of my group's ongoing work on data thinning. And this is really a story about a new way to split our data into independent training and test sets. So as any of us who works with data knows, there's many times that we'll be analyzing data, and what we need is to have an independent training and test set. So this comes up a lot, really, in almost any applied setting. But just sort of to help us think of some examples where we might want to do this. One example is maybe we are trying to fit a model to the training set, and we want to estimate its error on the test set. Well, it's really important to have an independent training and test set, because if we fit a complex model to the training set and then we try to estimate its error on the training, if we fit a model to the training set and we try to estimate its error on that same training set, then we're going to get a very poor estimate of the generalization error. So in this setting, we really want our training and test sets to be independent. Another example is maybe we have a really large data set and we're going to do a lot of exploration in order to identify a null hypothesis H naught that we want to test. Well, it's very important that we don't just generate H naught and also test H naught on the same data, because if we do that, we're going to get a very inflated measure of the significance of our results. So instead, typically what we might want to do in an applied setting is generate H naught on the training set and then test it on the test set. So these are just two canonical examples of applied settings in which it's very useful to have an independent training and test set. So of course, there's a strategy for generating independent training and test sets that's been around for a very long time that we're all very familiar with, which is sample splitting. And so the idea behind sample splitting is we have 5 observations, let's say, in the schematic. And what we're going to do is we're just going to split them into 2 parts, a training set, which in this case contains x1, x2, x3, and a test set, which in this case contains x4 and x5. And as long as my initial observations are independent, well, then x1 through x3 and also x4 through x5 will be independent. So we'll have independent training and test sets. And that means that I can, for example, fit a model on the training set and estimate its error on the test set, or generate a hypothesis on the training set and test it on the test set, and I'm good to go. So of course, sample splitting is extremely popular, and it's really like a key tool in the toolbox of any applied data analyst. Okay, so sample splitting seems pretty good, but it turns out it's not necessarily the right solution in all settings. So one example of when sample splitting might not be the right solution is when the sample size is small. So if we only have 8 observations, or in the example I just showed, 5 observations, it might be a tough sell to convince someone to split that very small number of observations into separate training and test sets, and then fit a model just on like half of those N observations. Another issue with sample splitting is that outliers will get assigned to either the training or the test set. And so if you have a couple of outliers in your data, then whether they get applied to either the training set or the test set will like really drastically affect your results. And so there's something pretty unsatisfactory about this. And most importantly, from my perspective, sample splitting doesn't always work. There are settings in which sample splitting is just not applicable. So one immediate way to see this is that we need the observations to be independent, right? So in order to guarantee that my training and test set are independent, we need the observations themselves to be independent. And that might not be the case, for example, for time series data or for data with spatial dependence. And then another setting in which sample splitting doesn't work is when the object of interest has length n. And this might sort of seem like a puzzling phrase, but it turns out that the application that I'll be talking about later, which actually motivated this work, which arises from the analysis of single-cell RNA sequencing data, is a setting where sample splitting doesn't work because the object of interest has length n. And I'll talk about that a little bit more at the end of the talk when we get to the application area. But the key point is that sample splitting is great. It's like such a wonderful tool for a lot of data analysis when it works, but there's cases where sample splitting doesn't work. And the goal of this talk is to find an alternative to sample splitting in cases where sample splitting is not an attractive choice. Okay, so again, today we'll be developing a new way to split our data into independent training and test sets. Okay, so our goal is something called data thinning, and the idea of data thinning is something like this. If I start out with x1 through x5, what I want to do is I want to generate actually 2 sets of data. One set is going to have a superscript 1 and the other set is going to have a superscript 2. And I want this to satisfy the following two properties. Well, rather, I want to be able to treat x1 as a training set and treat x2 as a test set. And in order to be able to do this, I would like x1 and x2 to be independent. So in other words, I want to be able to start out with my data and split it into x superscript 1 and x superscript 2 in a way where x superscript 1 can be my training set and x superscript 2 can be my test set. And so this is different from sample splitting because for sample splitting, we just put half of the observations in the training and half the observations in the test. Here instead, I'm splitting each observation in two. Okay, so we can write our goal a little bit more formally. So suppose that I have a random variable x that comes from a piece of theta distribution where theta is unknown. So the distributional family P is known, but the parameter theta is unknown. So for example, maybe P sub theta is like the Poisson distribution with parameter theta. And throughout this talk, theta is always going to be unknown. Anytime you see theta, that's an unknown parameter. Okay, and our goal is to generate x1 and x2 that satisfy two properties. I want x to be equal to some function t applied to x1 and x2. So here, capital T is a deterministic function. And if we think about this requirement that x equals t of x1, x2, what I'm saying is that I want to be able to recover x from x1 and x2. Once I have x1 and x2, I haven't lost any of the information in x because there's a mapping from x1 and x2 that'll take me back to x. And second, I want x1 and x2 to be independent. And this is important because I want to be able to treat x1 and x2 as training and test set. So I want them to be independent. And again, just to emphasize, the distribution of x involves an unknown parameter or perhaps multiple unknown parameters, which in my notation is going to be theta. And the fact that theta is unknown is really important because if there were no unknown parameters, then like we don't need to do statistics because we know everything already. So it's important that theta is unknown. Okay, so that sounds great. It sounds like a real dream. I also would like a unicorn. We can write down what we want to do, and the question is, do we actually know how to do it? So to sort of show you that this is something we should potentially be able to do, I'm going to return to sort of a very simple example that you probably did as a homework exercise in undergrad or in grad school, which is the Poisson example. So let's say that x is distributed Poisson theta. What I'm going to do is I'm going to generate x1 from a binomial distribution with size x and probability epsilon, where epsilon's a pre-specified number between 0 and 1. We can take it to be 1/2 in this talk. And then I'm going to take x2 to be x minus x1. And if this looks familiar to you, then that's because this is like the well-known binomial thinning of a Poisson random variable. And in particular, if I do this, then I get the following properties. First of all, x is equal to x1 plus x2 just by construction. And second, x1 and x2 are independent. And furthermore, just like by the way, I happen to know the marginal distributions of x1 and x2, and those are both Poisson with mean given by the original unknown parameter theta, but scaled by epsilon. So the key point to notice here is we started out with a Poisson random variable with some unknown mean theta, and we have a way to get x1 and x2 that are independent that we can sum to get the original x. And furthermore, doing this didn't require me to know theta, right? I was able to get x1 and x2 without knowing theta, even though theta is an unknown parameter, which is important because if doing this required me to know theta, then again, this wouldn't be useful. Okay, so this is sort of an example of the thinning that I want to do in this talk. But if you're a skeptic, you'll know, of course, that this is a very old result that's been in textbooks for many years. And so there's nothing new on this slide. Okay, so can we do this for a distribution that's not Poisson? So usually, if we're going to get something to work out, we should try on the Gaussian distribution. So now we're going to try this for Gaussian data with mean theta and variance sigma squared. So here the variance is known, but the mean is unknown. Okay. So what I'm going to do is I'm going to take x1 to be normally distributed with mean epsilon x and variance that's a function of sigma squared. Once again, epsilon is just a number that I get to choose between 0 and 1. We can just take epsilon to be 1/2. And I'm going to take x2 to be x minus x1. Okay, well, what do I get from doing this? Well, I get some really nice properties. The first property is just by construction that x equals x1 plus x2. The second property is that x1 and x2 are independent. And then also I happen to know the marginal distributions of x1 and x2, which are both normal with the same mean and variance that we started with, but up to a scaling by this parameter epsilon. And once again, doing this did not require me to know the unknown parameter theta. It required me to know the variance sigma squared, but it did not require me to know the unknown mean. Okay, so if you're going to be a skeptic, you might say a broken clock is right twice a day. So we managed to do this for the Poisson and Gaussian distribution, but is there a bigger principle at play here? Or is this just like a special case of two distributions that we can thin into independent training and test sets without knowing the parameter? And so the answer is there is a bigger principle at play, which I'm going to talk about now. But before that, I just want us to remember what our goal is. So our goal is that we're starting with x distributed p sub theta, where the distributional family p is known, but theta is an unknown parameter. And we want to generate x1 and x2 such that I can reconstruct x using x1 and x2 with some function t, and also x1 is independent of x2. And now I'm going to introduce a definition, which is the definition of data thinning. So consider a family of distributions p indexed by an unknown parameter theta. And I'm going to suppose that there exists a distribution g sub t, which does not depend on theta, and a deterministic function capital T such that when I sample x1 through xk from x, when I sample x1 through xk conditional on x from this distribution g sub x, where x follows the distribution p theta, then the following properties hold. The first property is that x1 through xk are mutually independent. And the second property is that I can reconstruct x from x1 through xk. And if I can do this, then I say that the distributional family P has been thinned by the function T. So this is my goal, is to find a way to thin a distribution P by a function T. So if I give you a distributional family P, if you give me a distributional family P, I want to be able to come up with a function T such that this definition is satisfied. So we can think about the Poisson example just to make sure we understand how that relates to this definition. Well, in this example, we're going to end up with x1 and x2 that are independent. So that satisfies the first part of definition one. And then the second part of definition one, to see that it's satisfied, remember that we can just sum up x1 and x2 to get x. So here the function capital T is addition. So just to understand this example, here my conditional distribution g sub t is just multinomial because the distribution of x1 and x2 conditional on x is just a multinomial distribution. And again, the function capital T is addition. Okay, so we can see that like binomial thinning of a Poisson random variable satisfies this definition of data thinning. One thing that's really important to notice is that this conditional distribution, g sub t, doesn't depend on theta. Again, because g sub t tells me how to get x1 and x2 conditional on x. And if that depended on theta, then this wouldn't be useful. Okay. So now we're going to look at the Poisson example again in even more detail, because this is going to help us develop a recipe to thin more general distributions. So it may seem like I'm going into much more detail that's needed on the binomial thinning example, but it's because we're setting the stage for a way to do this for other distributions that we haven't thinned before. So the way that I'm going to do this, I'm sort of going to work in reverse. Remember, my goal is that if you give me x distributed p sub theta, then I give you x1 and x2 that are independent. But we're going to work backwards. So instead, I'm going to say, just as a thought experiment, suppose that I have x1 and x2 that are independent Poisson random variables. Then I'm going to define g sub t to be the conditional distribution of x1 and x2, given their sum. By the way, that happens to be a multinomial distribution. And then I'm going to notice that x1 plus x2 is actually sufficient for theta in the joint distribution of x1 and x2. And so by definition of sufficiency, that guarantees that g sub t doesn't depend on theta. And finally, I'm going to notice that t of x1, x2, in other words, the sum of x1 plus x2 follows a Poisson distribution with parameter theta. And what that means, just because we've satisfied the definition of data thinning, what that means is that we have thinned the Poisson theta distribution by x1 plus x2. Or to state it in plain English, well, actually, I'll just state it in plain English. It's not on the slide. It's that if you give me a Poisson theta random variable, then I have a way to split it into x1 and x2 that are independent, such that x1 plus x2 sum to the original random variable. Okay, so now what about the Gaussian example? So in the Gaussian example, once again, we can see that we're going to sum x1 plus x2 to get x. So the function capital T is just addition. And now we can wonder, in this Gaussian example, what is this conditional distribution g sub x? Like, in other words, how are we sampling x1 and x2 from x? And the answer that we can see by looking at this is that x1 and x2 conditional on x follow a multivariate normal distribution. And in particular, this distribution of x1, x2 given x is just a singular multivariate normal given here. And again, as I mentioned, the function that we use to combine x1 and x2 into x is just addition. Okay, and we can just verify that this conditional distribution doesn't depend on theta. And so we're good. We've satisfied the definition of data thinning, and we can see that we've successfully thinned a Gaussian with unknown mean using the function addition. So now we're going to walk through sort of like the recipe to do this. The way that we come up with this example is we say, well, I want x1 and x2 to be independent normals with these means and variances. And then I'm going to define g sub t to be the conditional distribution of x1 and x2 given their sum. And then I'm going to notice that the sum is sufficient for theta. So what that means is that the conditional distribution does not depend on theta. And furthermore, the sum is itself distributed normal theta sigma squared. And so what that means is that I can thin this normal distribution with unknown mean using the addition function. Okay. So what's the recipe here? How would I thin an arbitrary distribution? Well, I would continue using this sort of backward seeming flow where I would start out by saying I want x1 to have a q theta 1 distribution and x2 to have a q theta 2 distribution where x1 and x2 are independent. And then I'm going to define t to be a sufficient statistic for theta in the joint distribution of x1 and x2. And I'm going to define g sub t to be the conditional distribution of x1 and x2 given the sufficient statistic. And by sufficiency, I know that this conditional distribution won't depend on theta. That's just the definition of sufficiency. And finally, I'm going to let p sub theta denote the distribution of the sufficient statistic. And what this means is that I can thin the distributional family p sub theta with theta unknown by the function t. Or stated in more simple English, if you give me x that follows a p sub theta distribution, then I can sample x1 and x2 from g sub x in order to get x1 and x2 that are independent. And by the way, x1 will follow q theta 1 and x2 will follow q theta 2. So again, this sort of might seem like a little bit like the orders backwards because I'm telling you that you need to start out knowing that you want x1 to be q theta 1 and x2 to be q theta 2. And then when you go through these steps, you end up finding out what the distribution is that you've thinned. It's sort of like you don't find out what you've thinned until the very last step when you figure out the distribution of the sufficient statistic. So you sort of have to start out with like a reasonable idea of what you want your x1 and x2 to be, and then you have to hope that the resulting p sub theta is a distribution that you care about. Because the p sub theta that you end up with, that's the distribution you've thinned. Okay, so this is our recipe, and I now want to try to convince you that this recipe is actually very general and that it can be used to thin just like a real diversity of distributions. And so to do this, well, first of all, the examples that I've already shown you have been all in the natural exponential family, and the function t has been addition. So I want to convince you about some examples where we're not in the natural exponential family and where the function t is not addition, so that you can see that this is sort of a useful approach that's very general. And so to come up with examples like this, what we're going to do is we're going to think about sufficiency. So a key idea here is that the thing that I'm thinning is t of x1, x2, where t of x1, x2 is a sufficient statistic for theta. So I'm going to organize my examples in terms of the notion of sufficiency. And to do this, I'm going to rely on some old results about sufficiency from the 1930s. So from the 1930s, we know that usually when we're talking about sufficient statistics, we're within the exponential family. But sufficient statistics also exist outside of the exponential family. And there's sort of two possibilities outside of the exponential family. Either the sufficient statistic grows in dimension with the sample size, or else we have a distribution where the support is actually governed by the parameter theta. So I'm going to use sort of these results about sufficiency in and out of the exponential family to organize my examples, where I'm going to first show you an example of an exponential family that's not a natural exponential family. So the Gaussian and Poisson were natural exponential families, but now we're going to look at a non-natural exponential family. And then I'm going to move outside of the exponential family, and I'll show you first an example of a family with support governed by theta, and then second, an example of a family where the sufficient statistic grows in dimension with n. Okay, so first, an example from a non-natural exponential family. And to do this, so remember, my recipe is a little funny because we're going to start out with x1 and x2, and it's not till you get to the end that you find out what distribution we're going to be thinning. So we can play a little game where I'm going to go through the recipe and you can see if you can guess what distribution I've thinned before I tell it to you. So my x1 and my x2 are both going to be beta, random variables with slightly different parameters. Remember, beta here is known, but theta in this talk is always an unknown parameter. And then I'm going to work out what is the sufficient statistic for theta, or rather a sufficient statistic for theta. And a sufficient statistic for theta is just the geometric mean, which is the square root of x1 times x2. And then I'm going to define g sub t to be the conditional distribution of x1 and x2, given the square root of x1 times x2. And by sufficiency, this doesn't depend on theta. And now I just need to find out what is the distribution of t of x1, x2. So if this were like a real life talk, then I would right now ask the audience to guess what is the distribution of t of x1, x2, and I bet some of you would have it right. But it turns out that the distribution of t of x1, x2 is actually a beta distribution with parameters theta and beta. So what this means is that I have a way that I can thin a beta distribution where the first parameter is unknown using the geometric mean. Or put that into plain English, if you give me a random variable x that has a beta, theta comma beta distribution, and you're interested in learning about theta, then I have a way that I can give you x1 and x2 that are independent and that both follow beta distributions involving that unknown parameter theta, so that you can fit a model to x1 and you can evaluate it on x2. Or you can generate a hypothesis on x1 and evaluate it on x2. Okay, so this was an example of an exponential family that's not a natural exponential family, and we can see that from the fact that the sufficient statistic was not addition here. The sufficient statistic was the geometric mean. So like it's a little funny because you're going to get this training set x1 and this test set x2, and they can give you back the original x, but you're not adding them. Which of course, like the beta has finite support, so you can't possibly add them to get back beta. Instead of adding them, you're going to take their geometric mean. Okay. So next I'm going to show you an example of a family with support governed by theta. So now we're outside of the exponential family, but we know that outside of the exponential family, sufficient statistics arise either in families with the support governed by theta or in settings where the sufficient statistic grows in dimension with n. So right now we're looking at that second case. So an example outside of the exponential family where the support is involving theta. So I'm going to take x1 to be a scaled beta random variable and x2 to also be a scaled beta random variable, where the scaling is by this parameter theta. So x1 and x2 are IID in this example, and feel free to try to guess ahead what distribution I'm going to be thinning here. So now I'm following my recipe, and I just have to work out the sufficient statistic for x1 and x2, or for theta with respect to x1 and x2. And the sufficient statistic is actually the maximum of x1 and x2. And now I'm going to define g sub t to be the conditional distribution of x1 and x2 given the maximum. And by the definition of sufficiency, this doesn't depend on theta. And so now I'm almost done. I just need to know what distribution the maximum of x1 and x2 follows. And it turns out that that distribution is uniform 0 theta. So in other words, I can thin a uniform 0 theta with theta unknown using the maximum. Or to put it in plain English, if you give me a random variable x that follows a uniform 0 theta distribution, I can sample x1 and x2 from gx to get x1 and x2 that are independent, where x1 and x2 each follow scaled betas, but involving that unknown parameter theta. And if I wanted to recombine x1 and x2 into x, I would use the maximum of x1 and x2. So again, if you have data that follows a uniform 0 theta distribution, and you want to have independent training and test sets, I can't give you independent training and test sets that are both uniform 0 theta. At least I don't know how. Maybe you know how, but I don't know how. But instead, I can give you a training set that's a scaled beta and also a test set that's a scaled beta, so that you can like fit a model or generate a hypothesis on the training set and then test it on the test set in order to do inference on data. Okay, and then the last example that I want to talk about is this example where we have a family where the sufficient statistic grows in dimension with n. And actually, in this example, what we're going to see is that sample splitting is a special case of data thinning. So we started off this talk saying that sample splitting is this really simple way to split data into a training set and a test set, and then we're going to develop an alternative strategy. Well, it turns out our alternative strategy is very general, and it's so general that sample splitting is a special case of this. So I'm going to illustrate that now. So in my previous examples, I had a distributional family P that was indexed by a parameter theta, but now instead I'm working in a potentially non-parametric family F. And in my previous examples, I just had like a random variable that I was working with, but now, or rather a single, I just had a single random variable. Now I'm going to have a vector. So I'm going to take x1 and x2 to be f to the n1 times f to the n2. In other words, x1 is a vector of n1 independent f distributed random variables, and x2 is a vector of n2 independent f distributed random variables, where f is some arbitrary family. I don't know what it is. I apologize. This sort of is bad notation because it looks like an f distribution, but here f is actually an arbitrary family. Okay, and I'm going to take s to be a function that sorts its arguments. So for example, if you give me 5, 1, 2, I'm going to give you back 1, 2, 5. All right, and now t is the function that concatenates and then sorts its elements. So like if you give me the vector 1, 4 and the vector 2, 5, then when I apply t to it, I'm going to give you back 1, 2, 4, 5. And so t is sufficient for f. And what do I mean by this? What I mean is that like if the data is x1 and x2, it doesn't actually matter like what's the order of the observations. All that matters is just like the set of observations concatenated and unordered. Right? If you have n1 draws from f and n2 draws from f and everything's independent, then the sufficient statistic is just like the set of draws. The actual order of the draws isn't important. Okay, so that's what I mean when I say that t is sufficient for f. All right, and now I'm going to define g sub t to be the conditional distribution of x1 and x2 given t of x1, x2. So what does this conditional distribution look like? Well, what we need to do when we think about this conditional distribution is we need to imagine drawing n1 independent elements and then n2 independent elements from a bucket of n1 plus n2 elements. So t of x1, x2 is just a bucket of n1 plus n2 elements because we've just concatenated and sorted all of the data. And so this conditional distribution involves just grabbing n1 elements at random and then grabbing another n2 elements, the remaining n2 elements at random. And that's exactly what we do when we do sample splitting, right? The idea behind sample splitting is we just have n observations and we just grab n1 at random, and then we grab the remaining n2. So what this means is that actually sample splitting is simply performing data thinning. We're thinning not the original data x, but just a sorted version of the original data x using the function t. where the function T just concatenates the training and test set, where X consists of IID draws from the distribution F. So stated another way, if you give me N IID draws from F, and you tell me that you'd like an independent training and test set consisting of N1 and N2 elements respectively, then of course I would just choose N1 elements and make that the training set, and then the remaining N2 elements would be the test set. But that actually fits directly into our definition of data thinning, where the function that we use to recombine the training and test set is just concatenation. And we can see that the conditional distribution doesn't depend on knowledge of F, because if I tell you we have a bucket of N1 plus N2 observations and please give me N1 at random, of course you don't need to know the family of those distributions to be able to do that. And so again, what's really nice is that this shows us that sample splitting is actually just a very particular special case of data thinning. And it also actually highlights for us, gives us a hint of when sample splitting might not be applicable, because we can see that in order for sample splitting to be a special case of data thinning, we needed our original data X to follow an FN distribution. In other words, we needed N IID draws from a distribution F. And so we can see that if we don't have independence, or if we don't have identical distributed random variables, then sample splitting will not actually come out of data thinning. And so that gives us a hint of when sample splitting might not be a good idea. Okay, so we have a main result in this paper, which is this theorem. But to be honest, I've already kind of said this verbally. But we can go through the theorem just quickly, but there's really nothing new that I haven't already said. So this theorem says that if I have a family of distributions P that's thinned by a function T, and we're going to let Q theta 1 through Q theta K denote the distribution of the mutually independent random variables sampled as in definition 1, well, then the following holds. First of all, T of X1 through Xk has to be a sufficient statistic for theta. And that's part A of theorem 1. So part A of theorem 1 says that T of X1, Xk is a sufficient statistic for theta. That's kind of obvious, because if T of X1 through Xk weren't sufficient for theta, then the conditional distribution G sub T in the definition 1 would depend on theta. So theorem 1 part A is kind of obvious, because in order for the conditional distribution that we're using to sample X1 through Xk to not depend on theta, we need it to be we need T of X1 through Xk to be sufficient. It almost follows just by definition of sufficiency. And then the second part of theorem 1 is also pretty straightforward at this point. It says that the distribution G sub T in definition 1 is the conditional distribution of X1 through Xk conditional on that sufficient statistic, where X1 through Xk are distributed Q theta 1 through Q theta K, respectively. So there's really almost nothing new in theorem 1. Theorem 1 is just telling us that if we're able to data thin, then that means that T was a sufficient statistic, and furthermore, that G sub T must have been the conditional distribution. Okay. So I've gone through a few examples of distributions we can thin, and you might be wondering, did I just choose like a few special cases? But the answer is no. Like, pretty systematically, we're able to thin a lot of distributions. So for example, in the natural exponential family, we can pretty much thin anything. And we can see sort of, if you look at the top of this this table in the natural exponential family, the sufficient statistic is going to be addition. So like, for example, to thin a binomial random variable, we're going to split it into multiple binomial random variables where the first parameter is scaled. And in order to get the original binomial from the thinned binomials, we're going to add them. Then we can move into the general exponential family, and I already mentioned the beta distribution and how we can thin that using the geometric mean. But we can also thin a gamma where the first parameter is unknown, a Weibel, a Pareto, a normal distribution where the variance is unknown. As I mentioned, sufficient statistics occur within exponential families and also in distributions with truncated support, where the support is governed by the parameter theta. And we already looked at the uniform zero theta distribution, but there's some other examples of families with truncated support that we can thin. And then finally, the non-parametric example that I talked about, where we assume that X follows an F to the N distribution. So in other words, our data is a random vector of length N, where each element is IID-F distributed. We can thin that as well. So essentially, like if you can come up with a distribution that involves a sufficient statistic, there's a good chance that you could thin it. We do have some examples of just of an example of a distribution that we can thin, and that's the Cauchy. So Cauchy is such a good counterexample for so many things, and in fact, we're not able to thin a Cauchy distribution. Okay, so we're really happy because our goal was to sort of come up with a framework that allows us to take a random variable with some unknown parameter theta and split it into multiple random variables that are independent without knowledge of that unknown parameter theta. And we've done it not for every distribution. For example, we can't do it for Cauchy, and another one we can't do it for is Bernoulli, but for a lot of distributions that are of interest. Okay, so now we're sort of done with like the methodology for today's talk, and just in the few minutes that I have remaining, I want to talk about an application. In particular, the application that I'm going to think about involves a case where I want to generate a null hypothesis on the training set and then test it on the test set. And this example is motivated by single cell RNA sequencing data, which is a relatively new, well, I shouldn't say new, but maybe a current technology that allows us to measure the expression of genes on individual cells. And so the typical pipeline for analyzing this data involves some sort of step where researchers identify latent structure in the data. For example, they might perform clustering of the cells, or maybe they learn a low-dimensional representation of the cells. And then they perform what's called differential expression analysis, where they want to test each gene's association with this latent structure. So for example, maybe they cluster the cells in order to identify subtypes of breast cancer or whatever. And then that's step one. And then in step two, they want to identify genes that are have different mean expression between the clusters that they've identified. And this two-step pipeline, this is used in like virtually every analysis of single cell RNA sequencing data. So here's an example. This is a Nature paper from 2019, and they released a Shiny app. And this is just literally a screenshot from their web app. So this is a very highly cited paper published in the top journal, Nature. And what they did was they clustered the cells, and then they have this little app that makes it easy to say that I want to look at clusters 11 and 14, which is shown in yellow and red. And then this app will actually compute p-values for you, testing for a difference in the expression of each gene between clusters 11 and 14. And those p-values are shown here in this table. And you can see there's some very small p-values like 10 to the -7 and 0. Okay, so this is like the state of the art in the analysis of single cell RNA sequencing data. So as a statistician, we might be concerned because the null hypothesis they're testing is that the expected expression for the jth gene is equal in clusters 11 and 14. But there's a problem, which is that we estimated these clusters from the data. And so we know that clusters 11 and 14 are going to look different on this data because we literally estimated these clusters on this data. In other words, we came up with this null hypothesis by looking at the data. And so there's a problem. Okay, so what can we do about it? Well, actually, first, just in case you're wondering if these p-values are valid, no, they're not. We can look at the software that's being used. So this is a software package called Seurat. This is like the state of the art. The screenshot says that it has 4,159 citations, but I actually took that screenshot a year or two ago, so it probably has a lot more now. This is a very well-known software, and it seems like the authors of the software know the problem. They said p-values should be interpreted cautiously, as the genes used for clustering are the same genes tested for differential expression. So it's like the authors of the software know that there's a problem with these p-values, but yet they're releasing them anyway. And of course, we as statisticians, we know what the issue is, which is that we need a training and a test set. Okay. Oops. All right, so is this just like a one-off problem? No, it's not. This is a paper published in 2020 called 11 Grand Challenges in Single-Cell Data Science. So that goes even beyond single-cell RNA sequencing. This is referring to an entire field of biology where they're measuring things on the level of single cells. And their challenge number 2 is that there's a problem if we don't propagate the uncertainty in the cluster assignments before we do differential testing between clusters. This is sort of biology speak for the exact problem that I just pointed out, where these researchers are doing clustering and then they're testing a null hypothesis of no difference between clusters, or alternatively, they're doing like PCA and testing a null hypothesis of no association between a feature and the principal component. So this is one of the 11 grand challenges in the entire field. So what is the actual issue? I know this is not news to any of you, so just very quickly, we'll sample 100 cells IID from a normal 0, 1. We cluster the cells, we compute the p-value for a difference in means, and all of those peerwise p-values are less than 10 to the negative 6 because we generated and tested the null hypothesis on the same data rather than having independent training and test sets. Okay, so we can try out sample splitting here. And what's interesting and sort of subtle, at least to me, when I first saw this, is that this is an example where sample splitting does not work. So let's just see what happened if we tried to apply sample splitting here. So I start out with some null data on the left. It's just normal 0, 1, no clusters. And I'm going to randomly split into training and test set, cluster the training set. Now I'm going to label the test set using the clusters from the training set. And there's a lot of ways you can do this, but to make this figure, I use 3 nearest neighbors classification. And now I'm going to test for a difference in means on the test set. So test for a difference in like the expected orange versus, or rather the mean orange and the mean green using like a 2-sample t-test or something. And if I do this, I get a really small p-value that's smaller than 10 to the negative 5. And when I first saw this, I was really confused because it's like, well, what's happening? This is null data. There's no clusters. So if I generate the null hypothesis on the training data, and then I test those clusters on the test data, it seems like since the training and the test data are independent, like my p-value should follow uniform 0, 1. And it doesn't. So what happened? And basically what happened here is that the act of coloring those test observations orange and green, even though I got the orange and green colors from the training data, in order to decide whether a given test observation should be orange or green, I had to use the test data for that. I had to use like the position of each test observation in XY space in order to decide whether to color it orange or green. So just the act of coloring my test observations orange or green actually used the data. So I've already actually used the data before testing the hypothesis on the test data. I've already used the test data twice, once to color the observations orange or green, and then to test whether there's a difference between the orange and the green. So this is an example where sample splitting doesn't work. And the way that I would summarize this really briefly is that sample splitting doesn't work in settings where the object of interest has length n. Like if I'm clustering n observations, I really care about that entire object of interest of length n. And if I split into a training set and test set, then using the training set, I can get like an object of length n over 2, but I needed the entire object of length n. And that's really where sample splitting gets into trouble. Okay. All right, so now finally, I'm going to talk about how we can combine these ideas in order to solve this open problem in single-cell RNA sequencing that involves doing clustering or learning a low-dimensional representation of our data, and then testing the null hypothesis that each gene is associated with the cluster or the low-dimensional representation. But we've seen that sample splitting won't work here, so we're instead going to use data thinning. And just a comment on what single-cell RNA sequencing data looks like. This is count-valued data. So we have a huge number of rows representing the cells in our data, maybe 10,000 cells, and then a lot of columns representing the genes, and there's 20,000 genes. And each element of the data matrix is a count. So it's a non-negative integer representing the number of copies of the transcript from that gene in that cell. So these are non-negative counts. And we're going to model these counts using the Poisson distribution. So we're going to use data thinning of a Poisson distribution. This is something that we refer to as count splitting in the context of single-cell RNA sequencing data. And so what's the idea? Well, we're going to take a particular element of this matrix. We're modeling it as a Poisson. So Poisson data thinning tells us to generate a training observation from a binomial distribution, and then generate a test observation by taking the original data and subtracting the training data. And we do this for every element in this data matrix to get independent training and test sets. And then we can fit a model on the training set and evaluate it on the test set. So that's the idea that we're going to apply, and we're going to apply it to some cardiomyocyte differentiation data in collaboration with our colleagues at Johns Hopkins University, Alexis Battle, and her PhD student, Joshua Pop. And so this data involves cardiomyocytes, which are cells in the heart. And this is cardiomyocyte differentiation data, which means that these are cells that are differentiating over time. So like they're starting on day 0, and then over the course of a couple of weeks, they're developing. So we're starting out with a certain type of cell that is like brand new, fresh out of the oven or whatever, and then it's developing into like a mature cell over the course of a couple of weeks. So on the basis of this data, the first thing that researchers do is project this very high dimensional data onto a couple of dimensions. And there's all kinds of different projections that researchers use in the single cell RNA sequencing field. But because this is a statistical audience, I'm just going to use PCA. So I'm just going to project the cells in this data onto the first couple principal components. This is a figure from their paper where each dot represents a cell, and they're using sort of the preferred type of projection that is used in their field called UMAP. But instead, I'm going to do PCA. And here's what I get if I do PCA. The first principal component is shown on the x-axis. The second principal component is shown on the y-axis. And here, each dot represents a cell. And I've colored the cell according to which day it is of this differentiation process. So day 0 is like a cell that's fresh out of the oven, and day 15 is a cell that's 15 days into its development. And what I can see is this clear gradient, like we have a rainbow going on here along the first principal component, where we can see that like the expression of these cells across the 15 days really does change over time in a pretty continuous way. So I can see with my eyeballs that like this first principal component is important. Like there's something going on, and I know that because it's very clearly associated with time. So I believe in my heart that I should find that there's a whole bunch of genes whose expression is associated with this first principal component. And so I can ask the question of which genes are associated with the first principal component, and I can do that in 2 ways. On the y-axis, I'm showing the naive p-values that I get if I perform PCA on all of the data, and then I test each gene for association with that first principal component on all of the data. So that's the y-axis, and I'm calling these the naive p-values. This is what I get, again, for my huge number of genes if I use the same data twice. First to generate the principal component, and then to test each gene for association with the principal component. So that's the y-axis. And on the x-axis is what I get if I perform data thinning, where I assume that my data follows a Poisson distribution. I thin it into independent training and test sets. I generate the principal components on the training data, and then I test hypotheses on the test set. So the 45-degree line is shown in black. So what I want you to notice is all of these p-values are small. If you look at the axes, like the axes go down to 10 to the negative 13. So there's a lot of small p-values either way. And that's really important because I believe that there are a lot of genes that are associated with this first principal component. So if data thinning like took away my power to identify the genes that are associated with this first principal component, then that would be really bad. So what this is saying is that all the p-values are small, or rather there's a lot of small p-values using either data thinning or the naive approach. So I haven't lost too much power. Although as you'd expect, the data thinning p-values tend to be a little bit bigger than the naive p-values, which makes sense because data thinning, like it's not overstating the level of association because it's successfully used in independent training and test set. But we might wonder, like, how can we explain sort of the amount by which data thinning is getting p-values that aren't as small? And to answer this, we can compute naive p-values using only the test data. So instead of computing the naive p-values on all the data, we can compute the naive p-values using only the test data. And in this case, the data thinning and naive p-values really fall on the 45-degree line. So what this is telling us is that in a case where the null hypothesis does not hold, the loss in power that we get by performing data thinning is entirely due to the fact the data thinning effectively has a smaller sample size. So data thinning, like it has the same number of observations, but there's less information in the test data because we've thinned the data. And so essentially, in a setting where there really is signal, so the null hypothesis really doesn't hold, we lose literally nothing by performing data thinning, other than just the fact that there's like less Fisher information in the test data than there was in the original data, basically. But now we might wonder, well, what happens if there's no signal in the data? Because here we had signal in the data. We know that there really were genes associated with the first principal component. But what if we're in a setting where there's no signal in the data, and then we would not want to have small p-values? We'd like our p-values to have uniform 0-1 distribution so that we know that the type 1 error is controlled. So to do this, we're going to take the same data, but we're going to only look at day 0. So this is just day 0. This is just cardiomyocytes that are fresh out of the oven that haven't had a chance to differentiate yet. And we have a couple of covariates associated with these cardiomyocytes, but we're going to regress them out so that our data just looks like a blob. And in fact, when I project this data onto the first and second principal components, I just get a total blob. So now I believe that there's no signal in this data. I think that these first and second principal components are just noise because these are all like identical cells all on the same day before they've had a chance to differentiate. And so I think that if we test each gene for association with that first principal component, like I think that those p-values should just be uniform 0-1. And so here's a QQ plot. So anything on the 45-degree line indicates like uniform 0-1 p-values. And then anything below the 45-degree line indicates an anti-conservative test with p-values that are too small. And so these are the p-values for those genes. And if I perform data thinning, then my p-values are right on the 45-degree line. So in other words, in this data that contains no signal, my p-values are exactly uniform 0-1 using data thinning. But if I apply the naive method that performs PCA on the data and then tests for association between each gene and that first principal component, then I get the green curve, which means that the p-values are too small, it's anti-conservative. Or the blue curve, if I just do this on the test data, also anti-conservative. So what I see is that when there's no signal in the data, data thinning, by giving me independent training and test sets, enables me to generate a hypothesis on the training set and test it on the test set without sort of using the data twice and obtaining invalid results. And so that's really great because we've shown that in the setting of single-cell RNA sequencing, we're able to maintain power in the presence of signal while not losing type 1 error control in the absence of signal. And just briefly to mention, in this example, I modeled the single-cell RNA sequencing data as Poisson, but there's been a lot of work indicating that maybe a negative binomial model is a better choice for single-cell RNA sequencing. And so in that case, I can apply the negative binomial data thinning that we've developed, which requires an estimate, or rather knowledge of the overdispersion parameter, which I would need to estimate in this case. So just to close, this work has been led by 2 wonderful PhD students, Anna Neufeld, who just finished her PhD at UW in statistics this past year, and also Amir Dharamshi, who's a current PhD student. The slide says first year, but actually he's a second year at UW, in collaboration with some really great collaborators, including Lucy Gao, who's my former PhD alum, who's now faculty at UBC Statistics, and then Jacob Bien, who's a longtime collaborator at USC. And here's our references. We have a whole line of work related to this, both sort of in the general setting and then in the specific instantiation of the application to single-cell RNA sequencing data. And we're really excited both about this work and also new work that's coming down the pipeline. So thank you so much for your attention, and I'd be happy to answer questions if there are any.
Okay, well, thank you very much for a very interesting talk. I think we should be clapping usually, but it's Zoom. So yes, exactly. Are there any questions from the audience? Yes. Martina?
Yes. Hello, can you hear me?
Mm-hmm.
Yeah, okay. So thank you very much for an amazing talk. I was just wondering whether it would be possible to say why you can't perform the data thinning for Cauchy, or I think you said Bernoulli. Like, what is the reason why it doesn't work?
Yeah, that's a great question. So the oops, I'm going the wrong way in my slides. So remember, one of the key things in data thinning was this notion of sufficiency, right? Where if I look at my recipe, I'm just trying to find my slide where I have the recipe for data thinning. I'm sorry if I'm making you all dizzy by clicking. But yeah, I need it. So I'm starting out with x1 and x2 that have this, q theta 1, q theta 2 distributions that are independent. And then I'm going to take t to be a sufficient statistic for theta. And then the thing that I'm ultimately going to thin is t. Or rather, if t follows a p theta distribution, the thing I'm ultimately going to thin is p theta. So the problem for the Cauchy distribution is that the Cauchy, there's no sufficient statistic, right? Like there's no way that I can reduce the Cauchy. And so I'm not able to get the sufficient statistic. So stated another way, the conditional distribution of x1 and x2, conditional on no matter what, is going to still involve theta. I just am not able to get rid of the theta.
Oh, okay, thank you.
Yeah. And then for the Bernoulli is a little bit funny, right? Because I can thin a binomial as long as the size is at least 2. But if I have a Bernoulli, then it's sort of like I just have 0 or 1. So I don't have a way to get two pieces from a Bernoulli that are independent. So we of course we have a formal proof of this, but the intuition is like if I have a Bernoulli random variable and it's just 0 or 1, like I don't know. It's just sort of like try to imagine splitting either a 0 or a 1 into two independent pieces that need to recombine in some way to get back that original 0 or 1. There just sort of like aren't enough dimensions of freedom there. And so we're not able to thin the Bernoulli. And we do have a proof of that. That's more articulate than what I just said.
Yeah, no, that makes sense. Thank you very much.
Thank you.
Skicha?
Yes, thank you for the great talk. First of all, I have just a short question. So in here, you have outlined the 1D case, and you have explained several cases of known families of distribution. However, my question is concerned with a more general case. So could this possibly be applicable to the vector random variables or to, let's say, matrix random variables? For example, do we know anything about the thinning of the Wishart distribution, let's say?
Yeah, great
you.
question. Yeah, wonderful question. Okay, so first of all, some of my examples here are vectors. So for example, I have like a multivariate normal in here, and a multinomial. The other thing is that, like, for example, in the Poisson example at the end with single cell RNA sequencing data, I had a matrix where I was modeling every element of the matrix as Poisson theta with its own, ij subscripts. So that was like a matrix of independent Poissons, which in order to thin a matrix of independent Poissons, I just thin each element independently. But the last part of your question was, so just to make sure I've answered the first part, there's no requirement here that X be a random, like a scalar random variable. X can definitely be a vector or a matrix. And in some of these examples here, X is a vector, but X could certainly also be a matrix. Furthermore, there's no requirement that theta be a scalar. Like theta could be multidimensional, and that's fine. Like all of these definitions and all this recipe still work out. The theorem still works out. The second question you asked was about Wishart. And the answer is yes, we can thin a Wishart, and it should be in this table. I think maybe we've sorted those details out maybe after I made this table. But sort of a way to think about it is the relevant line is right here. Oh, oops, I hope you can see my cursor. But we have normal mu theta with theta unknown. And then what we do is we notice that the sufficient statistic is x squared, which is the sum of the x's. And anyway, the point is we use the connection between a normal and a gamma with unknown variance. And so the Wishart is kind of the multidimensional version of that, where if I have like a multivariate normal distribution with mean vector theta and covariance matrix sigma, and then x transpose is Wishart, well, it turns out that we can thin it, and we can actually thin it by addition. And the algorithm for thinning it is relatively straightforward. So that was sort of a long way to say, yes, we do know how to thin a Wishart. So it's very cool, right? Because it means that if I have like some IID Gaussians where the covariance is unknown, then I can come up with a sample covariance for that, and then I can thin it into like sort of independent sample covariances that each are scaled versions of that original distribution of the covariance matrix. I hope that answer made sense, but the short version is yes, we can thin a Wishart. And we're actually, we have a paper that we're working on now where we talk about applying data thinning to dependent data like time series data. And one of the examples that we talk about is thinning a Wishart.
Sakicha, did that answer?
Yes, thank you very much.
Andre?
Thank you, Daniela. It was a great lecture, and naturally, I think opens up a lot of possibilities. But I have two questions. Firstly, there are cases, assumably, and there are, with multiple sufficient statistics for a case. How to separate thinned set based on made on different sufficient statistics for one case? Is there any test, and to separate which one performs best in certain cases? And second question, naturally, this is distributional. You mentioned many distributions. Could any of these be translated also to some non-parametric, possibly even semi-parametric situation? I think we have some measures of sufficiency also in those cases, how to transform it into non-distributional situations.
Yeah, that's a really good question. So the second question had to do with what about like a non-parametric or semi-parametric setting? So that's such a good question. I mean, the only framework in which we thought about that was in the connection to sample splitting, but I think it's quite likely that there's other ideas there and other work to be done, but we haven't done it yet, and I'd be really excited to hear anyone's thoughts about that. And then the other question involved the idea that like the sufficient statistic isn't unique. And so that sort of involves the, like the sufficient statistic is not unique, and which sufficient statistic should you be using? And it involves sort of the fact that this is such a backwards recipe, right? Like my end goal, like how did I come up with the idea that I wanted to start out with x1 and x2 being scaled betas? Like I don't care about the scaled beta. My goal was to thin a uniform 0 theta, right? But I kind of had to do a little bit of guess and check using the relationship between the uniform distribution and the beta distribution, and then thinking, well, maybe we can think about this as like theta times some beta. So maybe I need to have a theta times beta up here. And we kind of had to like just fiddle around to find a distribution of x1 and x2 that ended up giving us the uniform 0 theta. So you could ask, well, why is it max of x1 and x2 versus like the max of the square? Or rather, why is it the max of x1, x2 instead of the square root of the max of the x1, x2? Because that would also be a sufficient statistic. Like if the max is sufficient, then the square root of the max is also sufficient. And like the answer is because this was like the fiddling that we did that gave us what we wanted. So I don't have like a theorem that says that max of x1, x2 is like the best thing to do here. And maybe there's some other x1, x2 that I could have started with that would have also allowed me to thin the uniform. It's just that this is the one that I did that worked. So when I show you that big table of all these distributions that we can thin, in a way it looks like very polished and like all these different distributions. I've given you a recipe now to do it, but like under the hood, like the work that went into coming up with every row of that table was like literally just like guess and check, being like, okay, well, if I want uniform 0 theta, I bet that that's going to involve a beta. Let me just kind of goof around and like see what I need to do in order to get this to work out. And luckily for that, I have very talented grad students and collaborators.
Okay, thank you for a thorough reply. Thanks.
Tobias?
Yes, so again, thank you for the talk. This was super interesting. And also this sufficient statistic thing coming up was unexpected and I think very pleasant somehow. But so I would like to ask about this definition. You have, so as far as I could tell in all your examples, sort of motivated by wanting to have a training set and then a test set. So these are like two, you want to split the data into two parts. But in the definition, you have K parts. So is this just to be as generally as possible, or are there situations where you would want to thin it into very many parts? So how should we think about this?
Yeah,
K?
that's a great question. So yeah, I talk about training and test sets because it's a little bit easier to say verbally, but any of these thinning procedures, we could get K parts for like an arbitrary integer K greater than or equal to 2. And that might be useful, for example, if your goal is to like fit a complex model to the data and then obtain an estimate of the generalization error. If we just have a training and a test set, then that's okay, but it might be better to use like the analogy to cross-validation, right? Where we split our data, we thin our data into K thinned data sets, and then we fit a model to K-1, estimate the test error on the Kth, and then we repeat that iteratively in order to get like a higher quality estimate of the generalization error.
Yeah, okay, that's actually really interesting. I actually thought about asking about this cross-validation because this is sort of you can do this sample splitting, but there's sort of cross-validation. But I think I realized that this would have some of the same issues with the example you talked about, like doing PCA and then testing a difference in means. This sort of has the same issue. So I dropped
exactly.
this question.
So if sample splitting works, then you can use cross-validation instead of sample splitting. And similarly, if data thinning works, you can use sort of the cross-validated version of data thinning.
Yeah, okay. Well, very interesting. Thank you very much.
Thanks.
Then I think we have a comment or a question in the chat from Kimon. He's asking if you have any existing programs or packages available for data thinning, and
yeah, that's a great question. We do. For each of our papers, we have a software package, and it's linked to my website. I'm sorry that I don't have the URL right here, but it's just DanielaWITTEN.com. So my first and last name.com. And if you go to the publications and you look at the papers, then there's a software package for each. There's like really high quality tutorials. Well, I think that they're high quality tutorials, but if you disagree and you have any issues with them, please email me and we'll get it fixed. But yeah, I'd love for people to try it out. Please let me know any thoughts or questions you have about the software, because we really want this to be as useful as possible.
Are there any more questions? I may have a tiny quick question if you so I have also been working on this single cell RNA sequencing in the context of topological data analysis. I was wondering because in these methods you miss a little bit of information, if I understand correctly, in terms of shape, right? Because it's either clustering or PCA, so you make it linear in a way. I don't know if you had any thoughts about connecting data thinning as such with areas like.
So, Anne, that's a great question, but I don't actually care what kind of hypotheses you generate on the training data. So like if you're into like UMAP or like t-SNE or like any arbitrary, like nonlinear dimension reduction or topological data method or whatever that you're interested in, like you do that. Like please do that on the training set. Do whatever you want on the training set. I just need you to decide what hypothesis you're going to test based on the training set, and then we'll test it together on the test set. And since the training and test set are independent, and because the training set and test set both involved the same N observations, the same N cells, like we're good to go. So I just use PCA in that example because it's very familiar to statisticians, and I don't want people to get distracted by the details of UMAP or whatever, but it could be whatever dimension reduction you want. And it doesn't have to be dimension reduction, just anything that you're using to like whatever kind of complex modeling you're doing on your single cell RNA sequencing data. If you want to be able to test a hypothesis generated by that model. Just fit your model on the training set and then evaluate it on the test set.
I think my point is more is that you could generally you don't have this issue in terms of the small p-values if you take a slight different approach in that particular case.
I see. So that's interesting. So you're saying that if you do like a topological analysis or whatever, like a more complex method, then your is it like a dimension reduction? Like what is the method?
So you don't do a dimension reduction. You map it into a different space. So you don't do any reduction as such.
Yeah, but I would think that regardless of what it is you're doing, like you're mapping it into a different space, well, you're going to want to validate that mapping. You're going to want to make sure that it fits the data well. And to do that, you'll want to make sure it fits the data well. Yeah, go ahead.
For which there's a theory. No, but it doesn't rely, I'm saying it doesn't rely upon data thinning.
Right. Yeah, I'm just saying that in practice, like I think the most convincing thing for me is you know, if I have an independent training and test set, and so I feel confident that what I developed on the training set holds on the test set. And that's also the gold standard that like scientists are looking for.
Absolutely, but I'm not saying I'm saying bringing the two things together.
Yeah. Well, that sounds neat, and I'd love to read about that work.
Okay. Thank you. Any other questions? Yes, Kimon?
Yes, hello. Thank you for the presentation. Very interesting topic. My question is like very general. Do you have any assumptions that we have to fulfill in order to do data thinning, like linearity, multicollinear genes, or a sufficient number of observations?
Yeah, that's a great question. So the key assumption, and it's a critically important assumption, is that I know the distributional family. So like if I'm assuming that my data is negative binomial with an overdispersion of 3, but unknown mean, I need to be right about that. I need to be right that it's negative binomial with overdispersion 3. And if I'm wrong, then I'm not guaranteed independence of the training in the test set. So I'm actually making a very, very strong assumption that I know the distributional family. So that might seem really bad because that's such a strong assumption. Like when would I ever know that my data is actually
that was my second question.
Yeah, but I'll just say, like in statistics, nothing is free. Like it might seem like sample splitting avoids that type of restrictive assumption, but as we saw, sample splitting really requires IID observations in order for it to really fully make sense. And you know, IID observations, yeah, good luck.
Yeah, thank you.
Yeah.
Any other questions? I have one minor question. When it comes to wanting to apply this type of data to say where you indeed have dependence structures.
Yeah.
Is there anything you're already willing to say about some intuitive line of thought you have on that, or?
Yeah, I mean, and some of it is actually like it's sort of in this table. Like here we have a multivariate normal distribution with an arbitrary covariance.
Sure.
Where the covariance is known, and I can the mean is unknown, and I can thin it into multiple multivariate normals with the same mean and covariance up to a scaling. So here the covariance was known, but that has dependence. And then, yeah, I'm sorry, I'm not trying to be coy when I mentioned that we're working on this for correlated data as well. But I think you can imagine, let's say that instead I had normal 0 sigma, where now sigma is unknown. And so then the sample covariance matrix is going to then be Wishart sigma. And I can thin the Wishart just using exactly this recipe. And I'm sorry that the details aren't here, but the sufficient statistic will just be addition. And then I'm going to end up with just independent Wisharts that are each just like scaled versions of this. So it's really sort of exactly what you would think. And that's how Wishart thinning works. But unfortunately, I only know how to do Wishart thinning if I have more than one observation. So it's a little bit hard to say verbally. And if you send me an email, I'd be happy to sort of write it out for you. It's a little bit hard to say verbally, but just
Sure.
as I can't thin a Bernoulli, I
Yeah,
also
yeah, yeah.
can't thin a Wishart with just n equals 1.
No, sure not.
Yeah.